Today I'll be talking on the module polygenic inheritance and multiple allelism. So in this module we'll try to understand the basic concept of gene and alleles. Besides that, the main objective shall be understanding polygenic inheritance pattern, discontinuous variation and continuous variation, and explaining this continuous variation and continuous variation on the basis of or by putting or by fitting in a Punnett square test. Well, just to begin with, all the living organisms, irrespective of plants, animals, or human beings, have a number of physical traits. Say for plants, we may talk of shape of the seed, color of the seed, size of the seed, or maybe color of the flower, maybe color of the fruit, or maybe length of the stem, etc., etc. Similarly, while studying characters of human being, we may talk of skin color, then body weight, or stature, or yellow pattern, or blood pressure, pattern of our nose, hair colors, etc. So all these traits are inherited. So we study the inheritance patterns of such traits, and the study of such inheritance pattern is what we call genetics. That means genetics is the science of or the science which deals with inheritance of pattern from generation to generation. Now the question is, how inheritance is brought about? The answer is very simple. It is brought about by a unit of heredity, what we call genes, and what we call alleles. Just to have a fundamental concept of gene, a gene is particulate unit of heredity. It's a segment of DNA which is located at a specific locus in a chromosome. See, a specific locus in the chromosome, it's a very important point. The term gene was first used by Johansen in the year 1903. Now let us come to allele. Allele, on the other hand, is one of the several alternative forms of a gene occupying the same locus. That means at a particular locus, alleles always occur in a pair. One inherited from father and another from mother. So these two constitute together a gene. And the term allele was first coined by Batson. Now let us come to the traits which are controlled by alleles. Well, there are traits controlled by only one pair of alleles. That means two alleles. And the mode of inheritance of such trait controlled by two alleles or one pair of allele is what we call as simple single factor inheritance or single gene inheritance. As for example, the Yarlop pattern, the hairline, the number of digits, that means fingers. Such traits show discrete or discontinuous variation. That means, say as for example, in the earlobe pattern, the earlobe pattern may be either free or attached. Very discrete. No confusion. Whether free or attached. There are traits controlled by multiple alleles. Now here multiple alleles means genes which have more than 2 alleles, or rather polyallelic. That means there are more than 2 alleles but occupying the same locus of the chromosome. In other words, gene may have more than 2 alternative alleles but located at the same locus. Such traits also show discrete or discontinuous variation. Now, based on the number of alleles, number of genotypes may vary. So, if there are 2 allele, then it is called diallelic genes and can generate 3 genotypes. One which will be homozygous dominant, or maybe heterozygous, or maybe homozygous recessive. So that means diallelic genes can generate 3 genotypes. When the genes have 3 alleles, then it can generate 6 genotypes. 3 plus 2 plus 1. Then when genes have 4 alleles, it can generate 10 genotypes. Similarly, genes with 8 alleles can generate 36 genotypes. One of the very good example of multiple allelism is inheritance pattern of ABO blood group system. In this trait, human ABO blood group system, there are 3 alleles. They are A, B and O. And these A, B and O can generate 4 phenotypes. That is, whether the individual is either A blood group or B blood group or O blood group or AB blood group. So it generates 4 genotypes. On the other hand, it can generate 6 genotypes. Those 6 genotypes are whether the genotype may be AA, which will give phenotype A blood group, or maybe AO, which will give phenotype A, or BB, which will give phenotype B, or BO, which will give again phenotype B, or AB, which will give phenotype AB, and finally OO, which will give phenotype O. Therefore, different combination of these A, B and O alleles, which may occur at the same locus, of the chromosome can give 4 different phenotypes and 6 different genotypes. The early Mendelian genetics actually focused on very simple genetic traits which could be explained by single gene. That means a pair of alleles. For example, hairline. One may be either widow's peak or straight hairline. There is no intermediate. As I told you, it is discrete variation. Either this or that, no intermediate. However, by the early 20th century, most traits are far too complex to be determined by a single gene. And the idea of polygenic inheritance was thus born. Polygenic. Poly means many, genic from gene, polygene, many genes. So the term polygenic inheritance is used to refer to inheritance of quantitative traits. Remember, quantitative traits, which are influenced by multiple genes. They are controlled by multiple genes, not only one gene, multiple genes. In addition to involving multiple genes, polygenic inheritance also look at the role of environment in someone's development. That means in polygenic inheritance, there is also a role to be played by environment. This involves the inheritance. This means polygenic inheritance involves the inheritance and expression of phenotype being determined by many genes, but those genes located at different loci. Unlike those many alleles which occur at the same locus in case of multiple alleles. That means here in polygenic inheritance, it involves many genes. That means many alleles. And all these many alleles occur at different loci. And each one of the gene exerts a small additive effect in a continuous distribution mode. Here, no one single gene is dominant or recessive to another. That means effect of genes are cumulative. No one gene is dominant or recessive to another. That means effect of genes are cumulative, with each gene contributing a small amount to the final expressed phenotypes. Therefore, polygenic inheritance refers to a single characteristics that is controlled by more than 2 genes. It's also otherwise called multifactorial inheritance. So, a single characteristics that is controlled by more than 2 genes. Here, the more the number of genes controlling the trait, the more the number of phenotypes. So, there is a continuous variation. The more the number of genes controlling that particular trait, there will be more number of expression, more number of phenotypes. So, it shows a continuous variation. So, there will be naturally, the character will be characterized by a range of differences from one extreme to another. As for example, when we talk of our stature. The range of stature in normal individual may range from 125 centimeter to 200 centimeter. In between, one may have 130 centimeter, 140 centimeter, 145 centimeter, 170 centimeter, like that. So it shows a continuous variation. Now, polygenic inheritance occurs when characteristics are controlled by the alleles of more than one gene. Here, let us take some example. In human beings, the blood pressure, dermatoglyphics, Rees pattern, Rees count, circumference of head, human stature, intelligence pattern, skin color, heart diseases, body weight. These are some of the examples which shows polygenic inheritance pattern. Now here I shall explain you in brief the inheritance pattern of skin color. That means skin color is one of the best example of explaining polygenic inheritance. We know that human skin color is because of presence of melanin pigment. So the higher the quantity of melanin pigment in our skin, the darker the skin color will be. The more the melanin pigment, the darker the skin color. And the production of melanin pigment, on the other hand, is controlled by gene. We know that more melanin pigment, more darker skin color. But production of melanin pigment is controlled by gene. So assuming that three genes means 6 alleles occurring at 3 loci. I told you in polygenic traits, alleles occur at different loci, not at the same loci. In multiple alleles, it occurs at the same locus. So assuming that 3 genes, 6 alleles occurring at 3 loci controls the melanin production, let us assume that the 3 dominant capital letters, that is capital A, capital B and capital C, controls dark pigmentation because more melanin is produced by capital A, capital B and capital C. On the other hand, the recessive alleles of these 3 genes, that is small a, small b and small c, controls the light pigmentation because lower amount of melanin are produced by these alleles. Then finally, a genotype with all dominant capitals, that means capital A, capital A, capital B, capital B, capital C, the 6 alleles which occur at 3 different loci, has the maximum amount of melanin and will have a very dark skin color. On the other hand, the genotype with all recessive small case or small letter genes represented by small a, small b, small c, has the lowest amount of melanin and very light skin color. Now, further assuming that a male with a genotype capital A, capital B, capital B, capital C, capital C. That means having dark skin color, very dark skin color, marries with a lady, a female, having the genotype small a, small b, small b, small c. So when they get married, the gamete produced by male will be capital A, capital B, and capital C only. He cannot produce small a, small b, or small c because his genotype has only capital A, capital B, capital B, and capital C. Similarly, the female can produce the gamete having small a or represented by small a, small b, and small c. So this is a stage of gamete formation. Now, when the gamete fuse together, to form the zygote, the F1 generation offspring will have capital A, capital B, small b, capital C, small c. So a cross between a white gentleman and a black lady. So the result, the characteristics, skin characteristics of the offspring will be capital A, capital B, small b, capital C, small c. That means carrying 3 alleles for dark skin color and 3 alleles for light skin color. Now, when such two individuals carrying capital A, capital B, small b, capital C, small c get married, when they get married, then the possible gametes are 8. Capital A, capital B, capital C. This is one possible gamete of the male. Similarly, capital A, small b, small c. Then capital A, small b, capital C. Capital A, capital B, small c. Small a, capital B, capital C. Then here again, small a, capital B, and small c. Small a, small b, capital C. And finally, small a, small b, and small c. So these are the 8 possible gametes that can be produced by male. And the same set of gametes can be produced by female. So when these gametes fuse together to form zygote, there may be 64 possible offspring. 64 possible offspring with 7 different phenotypes. Now, these phenotypes are in such a manner that a particular phenotype may have no capital letter in each genotype A, B, C. That means it will be small a, small b, small c, small c. And one type of phenotype may have only one capital letter, and there will be such 6 cases, such 6 individual with or 6 individual having only one capital letter genotype. And there may be as many as 15 individuals with 2 capital letter genotype. And there may be 20 individuals with 3 capital letter genotype. Similarly, there may be 15 individuals with 4 capital letter genotype. Likewise, there may be 6 individuals with 5 capital letter genotype. And there may also be only one individual having all the 6 alleles represented by capital letters. So then this crossing between 2 individuals, a male and a female, having capital A, small a, capital B, small b, capital C, small c, with a female of same genotype will give 7 different phenotypic skin color sets. This can be very easily exemplified if we take it into a Punnett square. Now let us look into the Punnett square. Here in the Punnett square, the male gametes are kept horizontally and female gametes are kept vertically. That means in the column. See, the first gamete produced by male is capital A, capital B and capital C. So this gamete produced by male fuses with gamete produced by female, that too capital A, capital B, capital C. So ultimately the genotype will be capital A, capital A, capital B, capital B, capital C and capital C, which will represent extreme dark skin color. Then let us go to the next column, that is male gamete. Next male gamete, capital A, capital B and small c. It combines or it fuses with the female gamete capital A, capital B and capital C. Then it forms an offspring with a genotype capital A, capital A, capital B, capital B, capital C and small c. Likewise, going to the third column of male gamete, the gamete is capital A, small b and capital C. When it fuses with capital A, capital B, capital C of female gamete, it will form capital A, capital B, small b, capital C and capital C. Thus, it will have how many capital letters? It will have 1, 2, 3, 4, 5 capital letters. Coming to the fourth column again, first row, fourth column, it will be the fusion of male gamete capital A, small b, small c to be fertilized with capital A, capital B and capital C of female gamete. Then it will form capital A, capital B, small b, capital C and small c. So it will have 4 capital letter genotype. So likewise, when we keep on crossing, or when we keep on fusing male gametes with all the possible female gametes, there will be 64 products. And 64 product, as I told you earlier, it will give different 7 sets of skin color. This is how inheritance of polygenic traits can be simply explained using a Punnett square. So, we have understood what this continuous variation mean. Similarly, we have also understood continuous variation. This continuous variation are those discrete characteristics. Continuous variations are those having quantitative expression. And we have also come to know the difference between single gene and polygenic inheritance. At the same time, we have described the relationship between the number of genes controlling a character and number of phenotypes. We have also learned how to use Punnett square to work out possible genotypes of offspring for polygenic inheritance. Likewise, we can take into consideration of a number of polygenic traits and look the inheritance pattern of such polygenic traits.